Hello all, welcome to Life Science Library. In this video, we are going to see about next secondary metabolite, that is glycosides. It belongs to secondary metabolite category in Plant Physiology Unit 6 of CSIR Net Life Science portion. First, let us understand the basic details of what we are going to see in this video. We are going to see about glycosides, about saponins, about cardiac glycosides and cyanogenic glycosides. Tannins will not be included here. Tannins will be included in phenols, in polyphenols. Saponins, cardiac glycosides, cyanogenic glycosides, and the basic introduction of the glycosides are the agenda of this video. Tannin will be included in phenols. First up, we are going to see what are glycosides. Glycoside is a molecule in which carbohydrate or a sugar compound is being attached to non-carbohydrate moiety. Non-carbohydrate moiety by the help of glycosidic bond. Sugar being attached to non-carbohydrate moiety with the help of glycosidic bond. The non-carbohydrate moiety is also known as a glycon. What are the non-carbohydrate moiety? It can be terpene, it can be steroid, it can be flavonoid, it can be phenol. This is the basic introduction of glycosides. And it also contains a hydroxyl group. First, we are going to see about saponins, or glycosides containing steroids or triterpenoid aglycone linked to sugar by glycosidic bond. Here the aglycone component is steroid or triterpenoid. It can be both lipid soluble and fat soluble. Saponins are derived from saponaria or soapwort. It is employed as soap substitute. Saponin protects the plants against fungus and herbivores. It also has a great medicinal value of anti-inflammatory. The key here is aglycone component is steroid triterpenoid is the key point here. Lipid and fat soluble. Next, about cardiac glycosides or steroid glycosides, a glycone compound, which contain steroid nucleus linked with unsaturated lactose at 17th carbon. Example, digoxin is a cardiac glycoside. It inhibits sodium-potassium pump and increases intracellular sodium concentration. This is the example. So cardiac glycosides are steroid glycosides, a glycone compound, which contain a steroid nucleus linked with unsaturated lactose. Next we are going to see about cyanogenic glycosides. These are the derivative of 2-hydroxynitrile. Cyanogens are a glycone with cyanides to form glycosides with glucose. Cyanogenic glycosides are derived from L-amino acid of phenylalanine, tyrosine, valine, isoleucine. It is a natural source of hydrogen cyanide. The breakdown of cyanogenic glycosides releases HCN. So it is a natural source of HCN. It inhibits the activity of iron and cytochrome oxidase in electron transport chain. The cyanogenic glycosides inhibit iron and cytochrome oxidase. Now we are going to see about the structure of cyanogenic glycosides. R group linked to carbon, second alkyl group, oxygen linked with sugar, then carbon with 3 bond in N. This is the structure of. So we have a carbon group in the center linked with 2 different alkyl group, then oxygen, then carbon linked with sugar, then with nitrogen. R represents alkyl or aryl groups. This is the example of cyanogenic glycosides. This is the overall idea about glycosides. Let us sum up what are glycosides. Glycosides are carbohydrate groups, that are sugar groups that are linked with a glycone compound. This a glycone compound can be any carbohydrate sources. If it is steroid, or if the a glycone compound is steroid or terpenoid, it is known as saponins. The source of saponin is saponaria or soapwort. It is both lipid soluble and fat soluble. Next, cardiac glycosides. Cardiac glycosides in which they are linked to lactone, unsaturated lactose. Steroid nucleus are linked with unsaturated lactose. Other things are linked with glucose. Here the sugar compound is lactose. This is important. At 17th carbon, steroid compound. Here they are linked with glucose. Cardiac glycosides, they are linked with cardiac glycosides. They are linked with steroid, linked with unsaturated lactose at carbon 17. Example, digitoxin. It inhibits sodium potassium pump. Cyanogenic glycosides, they inhibit iron, they inhibit cytochrome oxidase. They are derived from L-amino acids, phenylalanine, tyrosine, valine, isoleucine, and source of HCN. Cyanogen is an A glycone with cyanide group. They are linked with glucose with glycosidic bond. They are linked glucose with glycosidic bond. This is the example of cyanogenic glycosides. Structure of cyanogenic glycosides, we have 2 R groups, O group attached with sugar, and another carbon group attached with N. The structure of cyanogenic glycosides. In short, glycosides, a glycon group linked with carbohydrate group with the help of glycosidic bond. With one exception, all other carbohydrates are D sugar, cardiac glycosides attached with lactone. This is an exception. Hope this video is clear. In this video we have seen about glycosides. Glycoside structure, saponin, saponin, cyanogenic glycosides, cardiac glycosides, and basic information about glycosides. Hope this video is helpful for everyone. Thank you for watching this video. Thank you.